The goal of my PhD research is to recapitulate the primary and metastatic microenvironments of human lung cancer in vitro. In vitro meaning that I will study the lung cancer outside its biological context, so not in a living system, as in an animal. In vitro models vary from shooting cell culture in a dish to elaborate 3D multicellular extracellular matrix gels. Alternatively, in vivo cancer models can involve implanting a human tumor into an immunodeficient mouse while the patient-derived xenograft, simulating a primary tumor, the subcutaneous injection of tumor cell lines, where the cell lines will then circulate in the bloodstream of the mouse, creating metastases. These models fail due to lack of human physiology in vivo and complexity in vitro. The success of future cancer models relies on bridging the gap between these current models. Thus, my research utilizes a platform which does just that: human organ on a chip. Organs on chips are microfluidic devices created with microfabrication techniques that contain hollow channels lined by living human cells that recapitulate organ-level structure and functions, for example, breathing in lung or flow through blood vessels. These chips are being developed to potentially replace animal testing for drug development and to create in vitro human disease models. Creation of tumor microenvironments with human cells, which also contain all of the physiologically relevant cues observed clinically, will yield a truly predictive model, benefiting patients and removing animals from the cancer research scene altogether. One of the major problems in the field of oncology today is that the same cancer in the same patient can often respond differently to the same chemotherapeutic agent, depending on whether it is located at its primary location or at a distant metastatic site. Multiple in vitro microfluidic models have been developed to investigate the mechanisms of cancer, as well as the metastatic cascade, including the study of tumor spheroids, tumor stromal interactions, angiogenesis, hypoxia, and intravasation under flow. Although these models have advanced cancer research and increased our understanding of the tumor microenvironment, none of these in vitro models permit analysis of how the cancer cells differ in response to therapy depending on their unique organ-specific microenvironment. My hypothesis is that the organ-on-chip approach, which allows us to recreate different types of human organ microenvironments, for example, lung versus liver, will enable me to develop in vitro cancer models that mimic organ-specific tumor responses to chemotherapy. This organ-on-a-chip modeling approach is exciting because it employs relevant human cells, overcomes disadvantages of current cancer model systems, and provides entirely new capabilities. If successful, this cancer on a chip will mimic patient response to therapy, proving the superiority of the human in vitro model to animal models since animal models fail to do so. I'm very appreciative to have received the graduate fellowship from the International Foundation for Ethical Research, and I look forward to making my contribution.